Hello everyone, welcome to J&J Mental Health webinars organized by Jonathan and Joshua Memorial Fund. Today, recipients of the Jonathan and Joshua Memorial Graduate Scholarships will be presenting their research. The Jonathan and Joshua Memorial Graduate Scholarships provides funding for a PhD scholarship in the area of mental health research in the Schulich School of Medicine and Dentistry. The scholarship will be awarded annually to a full-time PhD graduate student who is specializing in the neuroscience of mental health disorders and is not already funded by an external award. Let's welcome our speakers for today. Braden Terpu, Parachadat, Michael Mckinley, and Saloni Patel. So without any further delay, let's kickstart our session with our first speaker, Braden Terpu, MD, PhD student at Schulich School of Medicine and Dentistry. Over to you, Braden.
All right, hello everyone. As Hadi said, my name is Braden Terpu, and today I'll be presenting a series of research that I've titled collectively The Hijack Self. And specifically, we'll be looking at some altered activity as well as functional connectivity patterns that emerge in relation to a midbrain structure known as the periaqueductal gray. And specifically, we'll be looking at this in the context of post-traumatic stress disorder, or PTSD for short. So I'll start by having you imagine that you're on a run, perhaps a trail run. And as we're on this run, we're approaching some large boulder that's off the side of the trail. And as you get closer and closer to the border, imagine that out from pops behind it is a snake. So in this moment, we're probably going to feel as though there's this internal alarm system going off in our body, this rush of adrenaline, and we're snapped to the seat of our consciousness. But what exactly is facilitating or mediating this internal alarm response? Well, it's largely facilitated by different neurohormones that are released at the level of the brainstem and the midbrain primarily. So we're talking about subcortical structures. And what these neurohormones function to do is to shift our physiological state or shift our arousal state. We might have an increase in our heart rate, an increase in our muscle tone, and an increase in our respiration. And what these different physiological signatures help us to do is to respond effectively in turn. And when we respond, we may see ourselves take on one of a few different defensive phenotypes. These include fight, flight, and faint. So these defensive phenotypes are evolutionarily conserved, which means we see them across the animal kingdom. But what exactly is the neural circuitry that's mediating both the detection of threat-related stimuli as well as the subsequent response? Well, the innate alarm system is one such network. And what the innate alarm system refers to is this subcortical and interconnected network comprised by brainstem, midbrain, and thalamic nodes. The innate alarm system is centered on the superior colliculus, which lies in the midbrain. And the superior colliculus receives visual information directly from the retina. So in the context of our snake, this visual information would be relayed to the superior colliculus. So an interesting feature of the superior colliculus is that it's been shown able to differentiate between threat-related or neutral stimuli as they're presented in the outside world. So in the context of a snake, this would fit the bill of a threat-related stimulus. And then the superior colliculus would initiate this innate alarm system response by transmitting this information throughout this interconnected network. So the key function of the innate alarm system is that it can transmit threat-related information very rapidly. And the reason it can do this is because it bypasses a lot of the primary visual processing. So primary visual processing takes on the order of a couple hundred milliseconds, but in the context of a threat-related information, it doesn't necessarily matter how fine-grained the perception is. We don't need to know what color the snake is, for instance. All we need to know is that there's a snake present and we need to respond effectively in turn. And in addition to transmitting this threat-related information very rapidly, the innate alarm system also upregulates physiological arousal by stimulating or transmitting this information throughout the network. So I'll take a closer look at some of these innate alarm system structures. We have the superior colliculus, as I mentioned. It really centers the innate alarm system and it can initiate that response, predominantly involved in threat detection. We also have the amygdala, which is a limbic structure that's more so associated with emotion processing. So when you're experiencing fear or worry or anxiety, we often see some level of activity in the amygdala. In addition, we have the periaqueductal gray, which is a midbrain structure that lies just posterior or behind the superior colliculus. And whereas the amygdala is more so associated with emotion processing, so there's a conscious awareness to it that I feel or I am scared, the periaqueductal gray subserves more of the visceral and affective processing. So it's more so associated with these bodily sensations that represent fear or being scared. This I feel scared as opposed to I am scared, where really the conscious awareness or the conscious appraisal is what differentiates the two, emotion and affect. So when we look at the periaqueductal gray on this little slice here, we see that it has two dissociable subunits, the dorsolateral subunit and the ventrolateral subunit. And the interesting feature about the periaqueductal gray is that these two different subunits control the different arms of the autonomic nervous system. So whereas the dorsolateral subunit primarily modulates the sympathetic nervous system, the ventrolateral subunit predominantly modulates the parasympathetic nervous system. And together, the sympathetic and parasympathetic nervous system make up the autonomic nervous system. So if you electrically stimulate, for instance, the dorsolateral subunit of the periaqueductal gray in rodents, you will evoke a fight or flight response in the animal. Whereas if you electrically stimulate the ventrolateral subunit, you can evoke a freezing or a tonic immobility response. And we think of these as more affective shutdown responses. So I'll now quickly turn and discuss post-traumatic stress disorder quickly. So post-traumatic stress disorder has multiple different symptom patterns. We have the more traditional symptom pattern, and the traditional symptom patterns associated with are characterized by hyperarousal and hypervigilance mainly. This is the one we often see like portrayed in movies and media. These individuals often have recurrent flashbacks, nightmares associated with their trauma. And physiologically or viscerally, we see elevated heart rate, elevated blood pressure. And these can be recorded both during rest as well as during threat or trauma-related stimulus exposure. But there's a second symptom pattern known as the dissociative symptom pattern, which occurs in about 15 to 30% of PTSD cases. And it's characterized by much the opposite symptom profile. Instead of hyperarousal, we see hypoarousal, emotional blunting and numbing, and symptoms of depersonalization and derealization. And physiologically, we see reductions in heart rate, reductions in blood pressure in the context of threat or trauma-related processing. So what you might notice here between the traditional and the dissociative symptom pattern is that they're very akin to a sympathetic dominant and a parasympathetic dominant state, respectively. And since the periaqueductal gray coordinates the sympathetic and parasympathetic nervous system, that then puts the periaqueductal gray as a critical biomarker differentiating between these two different symptom patterns and just critical to the pathogenesis of PTSD more generally. So with that, one of my first studies was we wanted to assess this innate alarm system and specifically the subcortical correlates of the innate alarm system to see if we can find evidence that the innate alarm system is overactive or differentially active in individuals with PTSD as compared to healthy controls. So we had 20 individuals who met the criteria for a healthy control subject and 26 individuals who had post-traumatic stress disorder. And we asked all of the participants to generate trauma-related and neutral stimuli. So in the healthy control group, since they did not experience trauma, they were asked to generate material that was related to a strongly aversive or stressful memory that they had. What we're going to do is we're going to compare the responses to the trauma-related visual stimuli and the neutral visual stimuli. An important note here is that this stimuli will be presented subliminally, which means it's below the conscious threshold of awareness. So this is on the order of 16 milliseconds specifically, which is about enough time for you to know that you've seen something when you see it visually, but not enough time for you to differentiate whether you saw something that was trauma-related or neutral in origin. So again, just to repeat, the central question here is, can we find evidence that the innate alarm system is overactive in individuals with post-traumatic stress disorder? And we assessed this using fMRI. So when we looked at this, and this is the actual data displayed here on this glass brain, what we found was that in our PTSD group as greater than the healthy control group, we found this significant cluster of overactivation in the midbrain. Specifically, it was over top of the periaqueductal gray and the superior colliculus to a lesser extent. So that says that we did find some evidence, at least in the midbrain, that this innate alarm system is strongly overactive in individuals with PTSD. And I just repeat this on the next slide with a little bit clearer display. So in our PTSD sample, we saw stronger activity centered on the periaqueductal gray. And what this might suggest is that even though these participants aren't consciously aware that they're perceiving something that's trauma-related, they're still showing a differential response to the innate alarm system, which may then suggest that they're responding more defensively or engaging some physiological changes that they may not be perceptible. But this is just activity. So we looked at fMRI in the sense of activity in this first study. Next, we wanted to assess whether the periaqueductal gray is showing this activity as isolated or whether it's part of some larger network. And we can assess this by looking at the functional connectivity exhibited by the periaqueductal gray. So when you want to look at the functional connectivity of a specific set of voxels or a brain area, in this case, the periaqueductal gray, for all the participants, you can extract what's referred to as a seed time course or an activity profile. Essentially, this is just how this set of voxels changes in its activity over time, and time also marks the different onset of our stimulus conditions. But to save you a lot of the methodological jargon, essentially what we're looking for is this periaqueductal gray structure showing strong functional overlap with other areas of the brain? So we assess this using the psychophysiological interaction, and much like the previous study, we're going to compare the group differences between our PTSD and our healthy control group. But before I go into the details of what we found with this functional connectivity analysis, I quickly want to introduce this network known as the default mode network. So the default mode network is a large-scale intrinsic connectivity network that runs along the midline of the brain. So if you think about the midline, if you imagine that there was a brain in front of you and you were to separate it into its two dissociable or different hemispheres, you'd be kind of separating it along the midline. And all of those different cortical territories that stretch along the midline are often implicated within this network. It has critical hubs in the medial prefrontal cortex, for instance, as well as the posterior cingulate cortex and the precuneus, to name a few. And the default mode network is considered a resting state network, hence the name default mode. When it was first originated, the research around it, we thought of it as this resting state network because when you're asking participants to not actively engage in anything, this network would reliably come online, this cortical midline network. We've since expanded the definition of the default mode network to include internally directed cognition more generally, because the default mode network also shows recruitment, or it comes online during tasks like autobiographical memory. So when you're thinking about a memory that you've experienced from a first-person perspective, as well as self-related processing. So essentially, when you're thinking about your own thoughts, making self-other judgments, this network comes online. And an interesting feature about the default mode network is since it underlies both our memories, our firsthand memories, and self-related processing, it lays the foundation of sorts for self-experience or self-identity. This is really the network by which we experience selfhood, at least in part. So now transitioning back into what we found when we did the functional connectivity analysis. So in our PTSD group as greater than the healthy controls again, now we found that there was significantly stronger functional connectivity with the medial prefrontal cortex, as well as the posterior cingulate cortex and the precuneus, as demonstrated by these clusters of significant functional connectivity. So this tells us that the periaqueductal gray has really strong overlap functionally with two critical areas of the default mode network, the most critical areas of the default mode network. So again, I'll just show that data in a little bit easier diagram here. There's this weird reciprocal bidirectional functional connectivity exhibited between this subcortical structure, the periaqueductal gray, more so associated with defensive responding, and the network by which, or at least two critical nodes of the network, by which we experience self-identity or the sense of self. So this is critically important because we weren't expecting to find functional connectivity exhibited between the PAG and the default mode network. When we first sought out looking at the functional connectivity, what we thought we might see is that these downstream innate alarm system structures would come online. And the reason why this is so odd is because the default mode network is this internally cognitive network. So when we're thinking about our own thoughts in our head, whereas the periaqueductal gray is more so associated with external processing. I see a threat in the outside world, and now I'm looking to respond to that. And I'll quickly try to bring this all together in a nice little summary. So we have this midbrain structure that's associated with different physiological or visceral changes. It can increase or decrease our heart rate, blood pressure, our galvanic skin response, for instance. And this little midbrain structure is significantly overactive in individuals with PTSD, which might lead you to think that they're more defensively oriented, at least in the context of trauma-related cues. And individuals with PTSD often find trauma-related cues everywhere because of this attentional threat bias that we can discuss a little bit more in the discussion. But when we looked at the functional connectivity of this periaqueductal gray structure, we saw that it was really significantly connected to the default mode network, the network by which we experience our selfhood. And this has a lot of clinical interpretations. Could it be then that self-related processing, as subserved by the default mode network, is somewhat biased towards more threat or defensively oriented action, as subserved by the periaqueductal gray? There's a lot of different clinical interpretations that we offer with regard to these findings, and we've since expanded on them. And if anybody's interested in this research or wants to kind of see how it emerged and then kind of our interpretations of late, all of the research that I've discussed in this presentation is freely available on my ResearchGate account. You can just go in, quickly look at it. A big thanks to all the co-authors, of which all this work would not be able to have been done, and a special thanks to the Jonathan and Joshua Memorial Foundation. All of this research has been supported hugely and in part by the Jonathan and Joshua Memorial Foundation. So honestly, I can't say enough good things, and thank you so much.
Our next speaker is Faraj Haddad, MD, PhD student at Schulich School of Medicine and Dentistry. The mic is yours, Faraj.
Okay, hello everyone. So just a little correction, only a PhD, not an MD student. But today I'm going to tell you just very briefly about the work that I've been doing, and I'm going to go through sort of the idea of how infection during pregnancy can have some negative effects on brain development, and just how we study, excuse me, how we study this in the lab. Sorry. All right. So first off, I'm going to talk a little bit about brain development. So brain develops throughout our lives, starting from when we're very young and as a in the womb. So there's a lot of important steps that happen that set the foundation for the brain and how it's going to develop in later life. And even as young children, there's still brain development happening. A lot of this is these critical points where how we experience the world, whether we can see and hear, and all the things that we experience determine how our brain and our senses develop. And then later on, even in adolescence, it's not as much those basic functions that develop, but more so the connections between the different brain regions. And even as adults, our brain is never staying the same. It's called brain plasticity is a term given for it, and our brain's always changing based on our experiences and our emotions, our thoughts, and our feelings. And so today I'm going to tell you a little bit about this very early embryonic stage of brain development. And the reason I focus on this and why I'm really interested in it is because this is sort of like laying the foundation for the entire brain. So I really like this sort of Jenga tower analogy. So you can imagine if something goes wrong during brain development early on, it's like disrupting the initial foundation. And it's not to say that if you disrupt this foundation, that the whole tower is going to fall. But it might actually be more prone to falling if something else happens later on in life. And this is sort of the point that I want to emphasize throughout my presentation. And so we know from many studies that actually things that happen during pregnancy really affect the fetal brain. And these are sort of associated with factors, maternal factors, that we know pose a risk to the child. And so, for example, it's very well known that maternal smoking, alcohol consumption during pregnancy, and even using drugs or medications during pregnancy can have some very drastic effects on the offspring, right? And these are a lot of these are well known and it's common knowledge. But one thing which is where my field, where my studies go, is sort of looking at the effects of infection during pregnancy. And you can imagine this is actually pretty important now that we've been through a pandemic. And there's so many potentially pregnant mothers who have been exposed to COVID-19. And interestingly enough, this idea actually stemmed from epidemics and pandemics that occurred in the mid-20th century. And this is how even this whole field started. And so I'm just going to summarize quickly what we know from many human studies. And what we know is that fever, viral infection, and bacterial infection have all been associated. So if the mother has any of these three, there have been studies associating any of them during pregnancy with an increased risk of the child to develop autism or schizophrenia. Now, there are other associations with other disorders, but these are the two that have been most consistently reported. And so these are the ones that I'm going to focus on. Now, you're probably wondering, oh, I know someone, they had a fever or they had an infection during pregnancy. It's, you might be getting really scared right now because, I mean, infections happen on a regular basis. But really, what we know is that the risk increase, first of all, there's many things about this increase in risk of these disorders, which is that the risk increase is small but substantial. So it's not that as once someone, if a mom gets an infection during pregnancy, that her child is definitely going to get autism or schizophrenia. It's never black or white. It's always gray. But there is a risk, and it has been reported many times, and so we know that there is some factors that are important. Additionally, the risk is higher if the infection happens earlier. So sort of think about disrupting that foundation at a very early stage is worse than disrupting it at a later stage. It's worse if the infection is severe. So a lot of these studies that have reported the links have been looking at moms who have to go to the hospital for an infection. And also we know that a family history of psychiatric illness might contribute. So it's sort of, you know, you can have an infection and maybe that doesn't have an effect, nor usually, but if someone is predisposed through family history, then there might be more likely to be affected. Some other things that we know is that the infection type, it's not as important in this case where I'm talking about autism or schizophrenia. So whether it's a bacterial or a viral or a respiratory or a different organ infection, could be a urinary tract infection, it might not really matter for this. Any of these might increase the risk of autism or schizophrenia. This is different from other infections. You'll notice some other disorders like microcephaly, which is a big decrease in substantial decrease in the size of the whole brain. That, for example, has been linked with the Zika virus infection during pregnancy. But again, it's a different infection. What we're talking about is something that can be common to all of the different infections. But now you're wondering, well, how actually can it be common? Each of these different infections could be very different. So how do we link them together, and how can they be causing the same effect? Well, this is where the maternal immune response comes in. And so this is my analogy for what is actually happening. So once the body gets infected, it tries to clean up this infection. And on the way, it cleans up a little too hard and influences the fetal brain in the process. And so when the body gets infected with a virus or a bacteria, it produces these proteins called cytokines. And these are basically like, you know, the mop and the broom of our body, where they go, they influence the body to protect itself against infection. So they cause changes throughout the entire body. Now, the thing is, the body is used to, you know, it tries to use the same tools for different things because that makes things easier. And so it'll use the same cytokines to fight a bacterial infection or a viral infection. And so that's where the link comes in from different infections. And these cytokines, these proteins, can reach the fetal brain and can influence the brain development. And in the process, increase the risk for these developmental or neurodevelopmental disorders. And so now, how do we study this in rodents? So sort of what I do in the lab, obviously what we see the association in humans, but we want to understand what happens in rodents, what happens like at a more specific level. We have to use animal models. And so what we do is we mate rats in our lab, and then on a certain time during their pregnancy, we activate the maternal immune system in the mom. So some moms have their maternal immune system activated, and some not. And then we test their offspring for things that are related to autism or schizophrenia, right? And so you might be wondering, well, is there an autistic or a schizophrenic rat? And the real simple answer is no. Autism and schizophrenia are disorders that are, their diagnosis is really based on behavior. And a lot of these behaviors are very specific to humans. Social communication is very different in rats compared to humans. But we can actually study these disorders using animal models. And the way we do it in our lab is by testing this thing called the acoustic startle response. So this is what happens if someone, you know, pops a balloon behind you or slams the door really hard. You startle, your eyes blink, you kind of like shrug, and you get in this sort of, as Braden mentioned, sort of this response of fight or flight, but not exactly the same. And so this is a way we can study how the brain processes information coming to it, like sounds. So this is what it looks like in a rat. So this is a rat. Every time the X comes on, they're hearing a sound. In the human, you could see the eye blink, and you could see different rats. So this rat responds a certain way. This one doesn't really respond at all, and this one responds a lot more compared to the same sound. And so this test has been used extensively in research and actually helps us develop drugs for these disorders, and is especially for schizophrenia. There are some tests with the acoustic startle response that are staple tests for drug development at the early stages. And so what we know from many human studies is that individuals with these disorders experience sensory stimulation like sound or touch differently. So you might, hear a sound and react to it a certain way, but someone with autism or schizophrenia might hear it differently, and so their response will be very different. And so even though to you you're hearing a sound, it's not necessarily getting to the brain the same way for these individuals. And so just as a brief recap, what we see a lot of these changes in the rats that we expose to the maternal immune response during pregnancy. We see them having similar responses to individuals with autism and schizophrenia on this acoustic startle response. And so in the lab, our sort of broad goals for our research is to understand how these disorders develop. There's still a lot that we don't know. And maybe by understanding how this one particular risk factor leads to the development of certain symptoms related to these disorders, we can try to sort of put the puzzle pieces together and see what is actually causing these disorders. It also helps us understand how we can diagnose individuals more objectively. So the startle response is something that happens instinctively. You don't actually think about it. And one of the problems with autism, for example, is a lot of the ways we diagnose individuals is by getting their parents to report symptoms, which can be biased based on what the parent sees or what they would like to see. And finally, as I mentioned before, these are sort of like help us to develop treatments at the very early stages. So seeing whether something works and has potential to even be tried in humans and whether it is safe. And so with that, again, I'd like to thank you so much for all of your support. A lot of this research, once again, has been heavily supported by the Jonathan and Joshua Memorial Fund. And it's always been a pleasure working with Bob and everyone else. And this is a sort of a thank you from the Schmid Lab in one of our Zoom virtual lab meetings. And if you have any questions, you can learn more about our lab on this website. And if you have any questions, you can feel free to get in touch with me at any point as well.
Thank you so much, Faraj. Next, we have Michael Mckinley, PhD student at Schulich School of Medicine and Dentistry. Over to you, Michael.
Thank you. So my name is Mike, as I was introduced. I work in the neuroimaging and mental illness lab at Schulich. But for the past 6 years, I have been working within the first episode psychosis program at LHSC. I'm going to talk about a small section of my PhD work here today, but feel free to ask questions kind of broadly about psychosis, the program I work at, as well as anything specific you might want to know about my research in the follow-up questions. Just to give a quick background for those of you who are maybe unfamiliar with schizophrenia. Schizophrenia is a relatively rare disease, only impacting about 1% of the Canadian population. But at any time in Canada, 1 in 12 hospital beds are occupied by a person with schizophrenia, which suggests the burden to both the individuals and society broadly is pretty huge. And because of how chronic schizophrenia is, usually impacting people in the transition between adolescence and adulthood and carrying through the rest of their life, it's one of the top 10 causes of disability worldwide. Now, there are 3 symptom categories in schizophrenia. Positive symptoms are the symptoms that most people would be familiar with. These are hallucinations, most commonly auditory hallucinations or voice hearing, or delusions, which are just incorrect or beliefs that are not really grounded in reality. More interestingly to me are negative symptoms. So negative symptoms are the things we lose when we're diagnosed with schizophrenia. So social withdrawal, not seeing friends anymore, lack of emotional expression, and a number of other things that you can probably guess would really impact your life beyond your medical issue. If you're not able to maintain friends and a job, that can have pretty drastic impacts on your life. The final symptom domain is disorganization. So this is often depicted in movies and television shows in quite an unrealistic way, but this is when your speech, your thoughts, and your appearance, as well as certain specific cognitive domains, are disorganized, much like the name suggests. What's interesting about schizophrenia is that despite being a single illness, or theoretically a single illness, the degree to which people recover varies substantially. So this is a pretty good image, as it shows that the earlier you catch someone entering a psychotic episode, the more likely they are to respond well to treatment. However, the treatment remains really unpredictable for a lot of people. What's interesting, again, is that if you ask a patient what matters for them in terms of their recovery, they'll very rarely say the most important thing is not hearing voices or getting rid of their delusions. Most commonly, patients say basically what anybody in this room would say. They want to get a job, they want to go back to school, they want to have relationships, all of the things that makes life essentially worth living. And this is what are known as functional outcomes. So again, employment status, relationship status, and return to school and apprenticeship is what most patients are looking for. However, the sad story with schizophrenia is only 1 in 6 patients will ever return to their pre-illness level of functioning. And between 70 and 90% of patients are not engaged in meaningful work. So what this may mean they're on a disability pension for the rest of their life, as is the case with, in some samples, up to 50%. Or it may mean that they're unable to get a job that sustains independent living. In most cases, it's underemployment. So people with, let's say, a university degree or an apprenticeship that they've completed, but they're unable to work in their field because of their disability. In talking to patients at the clinic I work at, we really wanted to understand what predicts this difference in outcomes following diagnosis. And the reason we want to be able to predict this early in treatment is because the earlier we can identify someone who's likely to have a bad outcome, the earlier we can give them adjunct therapies. So typical treatment for schizophrenia is medication, antipsychotic medication. However, as I've hopefully made the case for, antipsychotic medications alone are often ineffective for a lot of the symptoms that people are dealing with. If we're able to identify those who are likely to do poorly on traditional treatments, we may be able to give them additional treatments like ECT, which, despite having a bad reputation from movies like One Flew Over the Cuckoo's Nest, is one of the most incredible treatments for people who don't respond to antipsychotics. And getting this done as soon as possible in treatment will allow patients to return to something resembling a normal life before they start missing major milestones. So what we wanted to look at primarily is brain folding in schizophrenia, so otherwise known as gyrification. The reason we look at gyrification is similar to a lot of the things Faraj has already talked about. Gyrification in adult humans is one of the best biological markers we have for early life developmental insults. So as you can see from this little picture at the bottom of the screen, it really hits its peak quite early in life. By about 2 months after birth, our brains are the most highly gyrified or highly folded that they'll be in their life. And this may matter for schizophrenia because in chronic samples, we can see that poor treatment response is associated with reduced folding. So these are the people who fail to return to work or don't do well on their initial medications. But in first episode psychosis or people really early in the disease, it looks like the flip might be true, suggesting that increased brain folding is actually associated with worsened symptoms. So we wanted to investigate this. To investigate this, I'm using the Tracking Outcomes in Psychosis study. This is a study I have been working on for a number of years now. And it's an MRI imaging study where we take patients when they first come into the emergency room or into the clinic before they've even started taking antipsychotic medications, and we scan them. The reason we scan them before they've started taking medications is most schizophrenia research has the issue that you're never sure if what you're looking at is the result of the illness or the result of the medication that the patient is taking. So for us, we don't have to worry about that problem, which is a huge advantage. The data I'm going to be talking about today is from roughly 60 of these first episode patients and about 30 healthy controls who are matched for age, gender, and parental socioeconomic status. So when we look at whether or not brain folding differs between healthy controls and patients, the answer is yes, but not to a great extent. Patients do show increased folding in an area known as a precentral gyrus, but how significant this is up to debate. What's more interesting to us is understanding the patients who do well versus the patients who are doing poorly. So when we restrict our analysis just to patients, you can see that in patients who are considered to be more severely ill, this is a combination of the psychiatrist's assessment, their social functioning, as well as their symptoms. There's increased folding in the patient's prefrontal cortex. This area is important for executive functioning and may be the reason that patients present atypically in the clinic if there's any sort of deformity in this region. When we look only at positive symptoms, so again, this is tying back to things like hallucinations and delusions, you can see that the areas that have increased folding or increased gyrification are a little bit more spread out throughout the brain. So a bunch of areas are impacted when we look at only positive symptoms. What's interesting is the area that I was looking at, negative symptoms, there's not that much difference in folding between the people who are doing really well and the people who aren't. There are a few areas that do pop up, as you can see here on the left side of the screen, but it's far less spread out throughout the brain than in other symptom types, which obviously wasn't what we were looking for initially. So in summary, patients do in fact have more highly folded brains in early psychosis, and among patients, more folding in the brain was associated with increased gyrification in multiple brain regions. But really what we're looking at doing is assessing how brain folding will predict treatment response and functional outcomes in our sample. So since that first batch of data was collected, I've been in the clinic several days a week following up these patients over the past year to 18 months. And we're going to be looking at if this folding pattern actually predicts outcomes that matter to patients. Now, the reason for this is all of those highly folded patients with more hallucinations and delusions that we showed you early, it may be likely that those people are more receptive to treatment. And the folks who didn't look as severely ill at onset may actually be the ones who do worse long term. So being able to tease those apart is an important next step before we can really start making a difference in how we approach clinical care. In summary, I just want to thank everybody who's involved with this research, and critically the Jonathan and Joshua Memorial Fund. Having this funding, despite how weird this year has been incredible in terms of maintaining productivity and being able to work with patients in the capacity that I'd hoped to. So thank you very much.
Thank you for that interesting presentation, Michael. Our final speaker is Saloni Patel, PhD student at Schulich School of Medicine and Dentistry. Saloni, floor is yours.
Hi, thank you. Okay, so my talk is titled How Loud is Too Loud? And I'll be going into specifically talking about hearing loss and the work I do in my PhD. So what is hearing loss? Hearing loss is the reduced ability to hear sounds, and about 1 in 3 people worldwide is affected by hearing loss, so it's quite prevalent. but a lot of them don't know it. So some of the signs and symptoms of hearing loss could include, difficulty understanding speech, speaking too loudly, and misunderstanding people. So if you or anyone around you, is constantly saying, can you turn the TV up, or can you repeat that? That's probably a good sign that, you should get your hearing checked. And how does this even happen, right? So causes of hearing loss include natural aging. So I'm sure many of you probably know over time, you do get hearing loss in old age, and this is just something we can't really control. It just happens. Specifically, like in the high-pitched sounds, you'll find go away faster. You could also, be exposed to or taking certain prescription drugs that can cause hearing loss. And what I'll be focusing on today is exposure to loud noise, which we actually specifically term noise-induced hearing loss. And the reason we care about this is because right now in the world, there are about 1.1 billion people at risk for this type of hearing loss. And it's honestly because of, certain popularities of certain types of things, or even like where you work. And to give you some examples is just, first of all, the most obvious, earphones. Things like AirPods and headphones have really become popular in the last, just decade, and people like to walk around with them and, blast the volume, and that's not really good for you. And then also going to concerts is pretty damaging. And then, if you're in the military or you work in construction or manufacturing, those are pretty loud environments as well. And, as you can imagine, over time, if you are exposed to this noise for months or even years on end, it could have a cumulative effect on your hearing. So why do we care about our hearing, right? Like, who cares if we have hearing loss? Well, to start off, hearing loss has been correlated with less education. So people think that this is because, if you're in class or even post-secondary education and you don't really understand what the professor or teacher is saying, you can't really work in groups because you can't hear that well, you're less likely to pursue more education or even do well in school unless you're, completely super motivated, like top 5% of students. And also that goes on into having limited job opportunities. So, less education, you're probably not going to go on and get more job opportunities. But also, even with a lot of education, you might not go on to get hired just because they'll have to accommodate you, or you might not be able to participate well in big group meetings and things like that. And lastly, just in your personal life, if you can't go to like a restaurant that's really loud and noisy and then talk to all your friends or the people you're going with, there is, some social withdrawal there, which can lead to isolation. And all in all, that could drop your self-esteem and confidence. So, quite a few different variables here about why we should care. For my PhD specifically, I wanted to look into this because just in the past couple of years, studies have found that hearing loss is actually the highest major risk factor for developing dementia. So I'd want to point out here that this is a modifiable risk factor. So for example, age is a risk factor for dementia, but you can't really stop aging, right? But hearing loss is modifiable. You don't have to have it. In most cases. So seeing that it is, the highest major risk factor for developing dementia, people start to ask questions there, like, because we've never really seen these two disorders linked at all ever before. So to get into why this could happen, well, some common things are, both disorders have been linked to increased inflammation in the brain, and they've also both been related to a decline in cognitive function. So some of these cognitive tasks, in humans at least, include mental status, some memory tests or executive function. And I've put these specific tests in brackets there if you want to look them up later. Just for fun, they're quite freely available. And a recent study also found that a mild hearing loss has been found to cognitively age the brain by approximately 7 years. So, let's say you're 43 years and you take a cognitive test. That basically means if you have hearing loss, then your test mark will look like what a 50-year-old normally gets. And, even though you're 43, if your brain is starting to perform at the level of a 50-year-old with a mild hearing loss, you can only, start to imagine what happens when you have a moderate or a severe hearing loss and how much your brain ages faster than your actual age there. So what we don't know is while both of these disorders show a clear evidence of cognitive decline, we have no idea how this happens. Like, we don't know what's going on in the brain. And some of the things that I ask for in my PhD is, in a comorbid model of both hearing loss and dementia, is there more inflammation in the brain? Like, is that, what's causing this? Or does hearing loss increase the amount of cognitive impairment you have if you then go on to develop dementia? So pretty much like these really big aims that I have for my PhD here. And since I'm kind of a newer PhD student, I can't really present my data just yet because it's unpublished, but I did want to go on and show you exactly how we can go on to study this in the lab. So as you've heard with some of the previous presentations, rodent models are quite popular for some of these disorders. And to study cognition, I'll show you an example of just one test of cognition. It's called the Morris-Water Mace, and it tests learning and memory. So if you look at this, it's a circular, tub right here. It's kind of like a hot tub size. It's a pool, it has water in it, and then it has these signs around. So there's like squares, triangles, etc. And then if you look at this circle right here, under the water, there's a little platform on which when rats go in here, they can stand. So it's like their own little swimming pool with a little platform where they can rest. And rats don't really like to swim. So as soon as we put them in this pool, they want to go around and find this platform. So I'll show you an example of that. Yeah, so as you can see, the rat there is swimming around, it's looking at the signs, and eventually it's going to find its way to the platform. It'll probably say, oh, look, the triangle is over here, and I remember it's by the triangle. Yeah, and as you can see, it's found the platform and it's done the test. Now what we do is we put the rat back in again, and we keep doing this. So over time, we expect the rats to learn, and remember where the platform is, which is by the triangle. So this way, we can track how fast they learn and also how well they remember their surroundings. Second, to study neuroinflammation or inflammation within the brain, we use some tests that are a little bit more translational. So neuroimaging, such as PET, MRI. I've included some, a picture here of what that could look like in a rat. And also immunohistochemistry, which is just a really, fancy word for we're going to dye the brain for specific inflammatory markers. So as you can see here, there's an outline of a specific area in the brain, and then all the little brown dots are just the immune molecule that we wanted to look at. Yeah, so all in all, that's pretty much what I do. A little bit more about me is I also did my undergrad at Western, and then I enrolled in the dual MD PhD program. So with, my long-term goal of being a neurologist that specializes in dementia and does, research in how to dementia prevention, I think that, scholarships and awards like the J&J helping someone through a path like this, or even, other students, it's very helpful. And honestly, it really moves the research forward. So that's good. Yeah, thank you for listening. And I think if you have any questions, please type them in the little question box there. And that's it from me.
Thank you, Saloni. Now it's time to answer your thoughts and questions. For those of you who want to ask questions using your own voice, kindly let us know in the chat box and use the raise your hand function, which is the hand icon on your right control panel now. Please ensure you have a working microphone for that. You can also type your questions in the questions panel, as I said before, which is located in the right corner of your screen. Our Q&A session will begin now. And I'm getting a good number of questions from the floor. I think, well, first I'm going to pass the floor to Dr. Andrew. He's one of our audiences who want to contribute to the panel. Dr. Andrew, I'm unmuting you. If you can, you just self-muted. Dr. Andrew, can you hear us?
Hi, can you hear me now?
Yes, clearly.
Perfect. No, thank you for having me on the panel. This is really exciting. I'm also a graduate of Western University and had previously received the Jonathan and Joshua Memorial Scholarship. So it's really exciting to hear about all the research going on at Western, particularly in Suzanne Schmidt's lab and Ruth Laney's lab, for example. And kind of one of the questions I had, as I'm a PTSD researcher and I work very closely with Braden, I was hoping, Braden, could you expand maybe on kind of, you were talking a lot about the default mode network and how trauma really becomes fused with someone's sense of self. I was wondering if you could kind of expand on that, given that post-traumatic stress disorder and trauma is often thought of as kind of a fear-based disorder. So I was wondering if you could kind of expand on, how you conceptualize trauma being integrated into the self and kind of being fused with self processes. I'd love to hear your thoughts on that. And thank you again, everyone. Your talks were very interesting, and yeah, I learned a lot. So thanks.
There's a lot to unpack there, Andrew. The first bit, I would say that for the long time, PTSD kind of got entrenched in a literature that was surrounding like fear circuitry, fear extinction, fear learning, and that's like a lot of the way in which we conceptualize post-traumatic stress disorder. But clinically, at least from patient accounts, this alteration to self-related processing has always been this lingering symptom that it's at least been of significant interest to me. There was a famous, not a famous paper, but a paper by Sutherland and Bryant, Sutherland and colleagues that I featured in a couple of my publications, in which they asked individuals with post-traumatic stress disorder to describe a memory in which there was the most self-defining for them. So you can think yourselves of a self-defining memory, but in the case of PTSD, what you often find is the memories that they're recalling or reporting are those that are trauma-related. So there seems to be this deep phenomenological or clinical link between self and trauma that appears in post-traumatic stress disorder. It's almost as though trauma deeply changes in some respect the way that you think about yourself, and you can't really separate the way you think about yourself from your trauma. When it comes to the neural correlates, as I mentioned before in the presentation, the default mode network is often this network by which we think that we engage selfhood or self-identity. And although the default mode network is predominantly thought to be this cortical network, some of the new area of research that we're investigating and you're very familiar with is to try to elaborate more on the subcortical components of these large-scale intrinsic connectivity networks. For a long period of time, PT or fMRI research was relatively limited to the cortex, just based on resolution practices. But now we're getting better and better at, we're getting stronger and stronger resolution. So it allows us to ask a little bit more nuanced questions. And with that, you can start asking questions about, how do these subcortical circuitries interfere or interact or interplay with these larger order networks like the default mode network. And that's where we're finding a lot of really interesting kind of bidirectional and in particular bottom-up connections. And as I discussed a little bit in this presentation, this PAG kind of showing the strong functional connectivity as well as directed functional connectivity kind of begs the question of to what extent are these subcortical systems that are more so associated with threat, fear responses involved in these larger scale networks. And I think I might have covered I think I covered most of what you're saying, but yeah, and with that, we're finding more and more that these subcortical components contribute a lot, and especially in the context of self, at least that's where my research is focused most extensively. Hopefully I got to the majority of what you asked.
Yeah, that's great. Thank you, Braden, for expanding on that. And just wants to say thank you to Jonathan Joshua Memorial Fund for putting this webinar together, as well as Bob McKinnon. And thank you for your continued support for all of our research in mental health. So thanks, and thanks, Braden.
Thank you, Dr. Andrew. We have another question, and that is to Faraj. Faraj, if there is no, well, I think you touched based on that and when you have your presentation, but I will gonna read the question as I have here. If there is no family history of autism or schizophrenia, does illness during pregnancy still pose a risk to get these diseases?
So the answer is yes. So again, as I said, it's a little bit complicated where it's not just this one factor, but people have found the risk to be increased even in larger populations, even when not considering family history of mental illness. And so it is still a risk factor, but as similar to Saloni's talk, it is sort of a modifiable risk factor, even though in this is because it is the maternal immune response. That is something that can be, you can reduce the impact of it even just by taking some medication. Although I don't want to sort of, there is very little that we know about whether that's something that works or not. So, but the thing I want to get across again is even without a family history, it could be a factor. And you also have to think that even those without a mental illness, they might actually have other factors. So despite the having no family history, there might be something in their early development in life which might make them more susceptible. And again, it might interact with these effects that happen during pregnancy.
Thank you, Faraj. I have a question to Saloni, and you pointed out, obviously, it was a great presentation about hearing loss. Saloni, I would like to thank you for that. And the quick question is, what medications contribute to hearing loss, Saloni?
Yeah, so I did mention in my presentation that was, one of the other causes besides noise. So there are quite a few actually that affect the ear, starting with antibiotic medications. So at a very young age, this usually happens to babies where they get bacterial infections, and then you kind of have to make like the executive decision, it's better if we give them these antibiotics that could potentially affect their ear. Because you want them to get better, like they're babies. So it's like a give and take there. And aside from young age, there's also, even like right now, I guess a more common one is aspirin. So aspirin has been known to induce hearing loss, and actually people, we don't do this in my lab, but people that study hearing loss actually give animals aspirin, just a lot of it, to induce the hearing loss. So it is toxic for your ear. And something that aspirin can also go into is if you have too much of it, the hearing loss also goes on to cause tinnitus, which is like a high-pitched noise that you hear, even though there's no noise, playing around you, which some of you might have heard of. So, quite a few drugs there that interact with hearing loss, but, really only makes a difference if you take too much of it. And then, or if you're sick. So some chemotherapeutic agents as well can also go on to cause hearing loss. But again, it's like that example I gave with the bacterial infections in babies, where you just gotta win some, you lose some, right? Like which kind of thing do you want to target, problem-wise in your body?
Thank you, Saloni. We're getting a good number of questions from the floor. Faraj, next is being addressed to you. It seems like it might be difficult to detect schizophrenia at the young age. Are there opportunity to detect anomalies in DNA, if that makes sense?
So so that's more of like a genetic. So if I'm understanding the question correctly, if you can detect something in the genes of someone, and that might indicate whether they have a higher risk of getting schizophrenia before they develop it. And the answer is yes. There are multiple genes that where if you have mutations in them, you might actually have a bigger chance. And this is also something where my work comes in. So people have shown in rodent models that a mouse, for example, who has one of these gene mutations that we know from humans is linked to schizophrenia, when they get this infection during pregnancy, the impact becomes worse. So more than just the infection and more than just the mutation itself. But yeah, to answer the question, there are genetic risk factors and changes in the DNA that we can detect, but it is difficult. So it's not always that if someone has it, that means they will develop schizophrenia. Yeah.
Thank you, Faraj. Getting another question from the floor, and that is to Saloni. Has it been ruled out that the same risk factors that cause hearing loss are causing the dementia instead of actual hearing loss? Thinking of industrial exposure and other things. Saloni?
Yeah, so just trying to understand the question, is it the things that cause hearing loss, is it that's contributing to dementia instead of the hearing loss itself? So studies in humans have actually shown that it's not specifically the risk factors for hearing loss. So like I mentioned, exposure to loud sounds, they found overall, it doesn't matter like why you have hearing loss. You could have it from a young age because of drugs, you could have it because you're exposed to noise. So, all very different scenarios that it in itself correlates with a higher risk for dementia. So yeah, it does sound kind of weird there, because I think even like myself growing up, I never associated the two at all, and I never thought that would ever be an association. But yeah, it is relatively a very new finding. So I think kind of digging into it and seeing what exactly the commonalities are there. I know I, again, didn't present anything that I've, found just because it is unpublished, but to kind of give a little bit of an idea. So hearing loss causes a very specific type of memory impairment, which is the same as the type of memory impairment you see in dementia, specifically Alzheimer's disease. So we think that maybe because they're, working on the same aspects of memory and cognition, that because, it's hitting the same thing, both of them together could have that worse effect. Yeah, I hope that helps.
Oh, certainly. Thank you for sharing your insights, Saloni. And I do have a question for Michael. If you can hear us, what age for first episode psychosis in schizophrenia? Michael?
So that's an interesting question. Most typically, we would expect the first episode to hit between about 16 and 24. And for males, it would be very rare to be much older than that. It can vary by a couple of years. With females, it's a slightly more complicated question. The most common time to have your first episode would be, again, that 16 to 24 kind of peak period. But there is a smaller subset of women that will go on to develop a psychotic episode right around the time of menopause. So kind of early to mid 40s is not unheard of among women, but it is very much unheard of for males.
Thank you very much. Yeah, I think I just want to thank you all once again. Thank you, speaker, for answering all the questions and taking the time to share your thoughts. Our special thank you to all the attendees who participated in this webinar. So until we meet again, stay safe and don't forget to check our website for more details on our upcoming events. Thank you. Have a good one.